Hello everyone. So today we are going to learn about microbial growth, part 1. Okay, so let us start. So microbial growth, depending upon the nutritional status, bacteria exhibit different growth patterns. Okay, so in laboratory, bacterial growth can be demonstrated by plotting a graph between microbial number and time duration, right? So in case of microbiology or the students of the microbiology, this growth study is a must topic for the students. Okay, so everybody should know about how to measure this growth of the bacteria, right? Now, how is microbial growth measured? Okay, so it is measured as optical density in spectrophotometer. Now, optical density is also sometimes referred to as absorbance. Okay, so this microbial growth results in turbidity, which is an index of bacterial growth. Right. So suppose we have done a fresh culture of some bacteria, and after incubation period, after 24 hours or 48 hours, whatever. So when you see after that incubation period, you will see that your transparent media has turned into turbid media. Okay. This turbidity is a sign that your bacteria has grown in your culture media. Okay. So this turbidity is an index of bacterial growth. This shows the bacterial growth. Now, the density of this suspension, it can be measured in spectrophotometer as absorbance or optical density. Why? Because this is directly proportional to the cell concentration. Okay, so the amount of turbidity can detect the number of cell present in the bacterial culture. Okay, it will not detect the exact cell concentration, but it will detect somewhat approximate cell concentration. Okay, so this is how the microbial growth is measured. Now, what is the principle of the absorbance or this optical density? The cells that are suspended in the culture, what they do, they interrupt the passage of the light. Now, if you see the structure of a spectrophotometer, you will find there's a light, there's a detector which detects the light that passes through these cells, and it will give you the optical density, right? So the cells suspended in the culture, they interrupt the passage of the light, allowing less light to reach the photoelectric cell. Now, this will what it will do, the amount of the light energy that gets transmitted through the suspension, it is measured as a percentage of transmission or as a optical density. You can do either. Okay, so here if you do as a transmission, then you have to set your blank to 0%, and if you are doing it with optical density, then you have to set your value to 0. Okay, so the amount of light that gets reflect from the cells, that will get measured, get detected by the detector which is present in the spectrophotometer, and this will give the value as optical density or transmission in the spectrophotometer. Okay, now this absorbance, this is a logarithmic value, and this is used as to plot a graph of bacterial growth. Suppose you are taking in transmission, you have to convert it to optical density only. So optical density or absorbance, this is a logarithmic value, and this is used to plot the graph of the bacterial growth. Okay. Now coming to the generation time. Okay. So before starting, we need to know the causes that affect the microbial growth, right? So warmth, moisture, pH levels, and oxygen levels, these are the four big physical and chemical factors that affect the microbial growth. Okay. Now coming to the generation time or doubling time. So this bacteria, they reproduce through a process called binary fission, right? So they reproduce, they do not do asexual reproduction, they do binary fission. And where in the binary fission what happens, one cell divides into two to form the next generation, right? Now the time difference between one generation of bacteria to double into two daughter cells is the definition of the generation time. That means generation time is the time difference between one generation to another next generation. Okay. Now this generation time is also known as doubling time. Now suppose you have one bacteria. Okay. When you put the fresh culture medium, you put some inoculum into it, right? So let us suppose that you have one bacteria in your culture, and this will grow in what way it will grow. So it will grow from 1, it will become 2, From 2 it will become 4. It will divide into in the form of uh, 2. Okay, it will increase in the fraction of 2 to the power n. We'll see it later. So from 4 it will divide into 8 cells. So this is how the bacteria cell divides. So from here you can see there's the 1 cell, 2 cells, 4 cells, 8 cells. Okay, so this is the first generation, second generation, third and fourth. So in this, from the second generation onwards, you can see that the cells are dividing in a fraction of 2 to the power n. Okay, now details of this division we'll study in our part 2, but for this you need to know that from 1 cell it divides into 2, and from 2 it divides into 4, 8, and this goes on in a fraction of 2 to the power n. Okay, now what are the microbial growth patterns that we know? So there are 4 phases or 4 patterns of microbial growth. First is lag phase, then log phase, then stationary phase, and then death phase. Now lag phase is what is lag phase? In a freshly inoculated broth media, bacteria take time to adjust in the new environment. Okay, this gap of time is called the lag phase. So you are giving a new bacteria, sorry, new culture media to the bacteria. So this bacteria, what it will do, it will take some time to get adjust in the environment. In order to, it prepares itself in order to reproduce. Okay, so this gap is called the lag phase. Now in the log phase, what happens? This bacteria is ready to divide. So what it does in log phase, it uses the nutrients of the media and multiply very fast, which shows the exponential growth. This in the log phase only, the bacteria grow in the exponential form. Okay, it increases exponentially. This period is called exponential phase or the log phase. Now after that, after some period, what happens? The growth becomes stagnant. Why? Because the cell growth stops. After log phase, what happens? The growth becomes stagnant because the cell growth stopped. But since the cell growth is stopped, it doesn't mean that they are not active. They are still metabolically active. Okay, but there is no increase or decrease in the number of microbial cell. That means what? They are not dividing. In the stationary phase, they are not dividing. Okay, so there is no increase or decrease in the number of the microbial cell in the stationary phase. So this is called the stationary phase. Now in the death phase, what happens? The nutrients of the medium starts to deplete. Okay, so since the nutrients of the medium are starting to deplete, and the bacteria is not dividing. Okay, the microbial cells are not dividing. So what happens? Continuous accumulation of the toxic metabolites occurs. Right, so toxic metabolites, they starts to accumulate, and therefore the death of the bacteria occurs. So that's why this is called death phase or decline phase. Okay, so these are the 4 phases, 4 growth patterns of the microbial culture. Now coming to the growth curve, if you see, so there's a x-axis and there's a y-axis. So in y-axis we plot the logarithm of living bacterial cells, or simply absorbance we can put, and in the y-axis we put time in minutes, in hours, whatever interval you take. Okay, so this first one is our lag phase, as you can see. Second one is our log phase, which is also known as exponential phase. Third one is our stationary phase, you can see it is linear. And the last one is our decline phase. Okay, decline phase or death phase. Now let us recall what we have learned. So lag phase is the time taken by the bacteria to adjust in the new environment. Right. What is log phase? Log phase is that the bacteria, they are using the nutrients of the medium and they are multiplying very fast. So in the log phase, the multiplication, as you can see, that it is increasing exponentially. Okay. So that means the bacteria is continuously using the nutrients of the media that you have given, and they are multiplying very fast. Now in the stationary phase, there is no increase or decrease in the number of microbial cell. Okay, so here you can see, therefore this is going in a stationary, it is a straight horizontal line, as you can see. Okay, so there is no increase or decrease in the number of microbial cells. Now the last one is our decline phase. Okay, last one is our decline phase. So in decline phase, this is the nutrients of the medium has started to deplete, and there is a continuous accumulation of the toxic metabolites. Therefore, the cells are starting to die out. Okay, but you can see here that this decline phase never reaches this x-axis. Okay, because some of the cells will remain. So the reviving process of the microbial cells are easy than the animal cells. Okay, because no matter what the condition, some of the bacteria, some of the cells will go in a dormant stage and they will remain alive. And when you do the reviving process, so that dormant cells will again get will again live. Okay, will again reproduce, will again do the division, and they will again be revived. Okay, so that is why this line of the decline phase never touches 0 or x-axis. Remember this one, this is a very important point to remember. Okay, now what are the types of growth curve, what we can see? Okay, so types of growth, there are four major types of growth curves which we see generally. So the first one is our exponential growth curve. Obviously this is the most common type of growth curve. Second is our linear growth curve. Third is our logarithmic growth curve. And fourth is our logistic growth curve. Okay, so these are the four major types of growth curve. Now, linear growth curve is also sometimes called as arithmetic growth curve. Okay, you should not get confused. Linear growth curve is also sometimes known as arithmetic growth curve, and this exponential growth curve is also sometimes known as geometric growth curve. Okay, so if the question comes like you are given a geometric growth curve, so you need to understand that they are talking about exponential growth curve only. Similarly, if it is written in the question that arithmetic growth curve, so that means they are talking about linear growth curve. Okay. Now, other than this, there are two other curves which we can see. That is sigmoidal growth curve. This is commonly seen in the batch culture, and this one is linear or straight growth curve, as we have studied in our second types of growth curve. So this is also very common, and this is mostly seen in the fed batch culture. Okay, so in total we can say there are five growth curves that we see commonly. Okay, now coming to the growth measurement, microbial growth measurement. So we need to measure the bacterial culture, right? So how we will grow, this is the requirements that we need for the microbial growth measurement are pure culture of the bacteria. You need a pure culture. Here we will take E. coli, example of E. coli will take. You need a suitable growth media, for example, nutrient broth media. You need a flask for the culture, spectrophotometer for reading or to taking the optical density, incubator and shaker. Okay, so coming to the procedure of measuring microbial growth. So how to measure? So in the first step, what you do, you prepare fresh microbial broth culture. Here we have taken E. coli. So you prepare fresh microbial broth culture and you incubate the culture till it reaches 1 into 10 to the power 6 cells per ml. Okay. So you need to have minimum of this 1 into 10 to the power 6 cells per ml. Right. Now, in the step 2, take 5 to 6 time intervals of 4 to 6 hour difference. For example, 0, 6, 12, 18, 24, 30 hours. So this is 6 hours difference. They have 6 hour difference between them. So you can take 5 to 6 time intervals of 4 to 6 hour difference. Okay, now here's a note, you can take the time intervals according to your requirements. If you want to take it at an interval of 24 hours, 48, 36, 72, you can take it. If you want to take it in a difference of 4 hours or 3 hours, you can also do that. So you can do it according to your requirements. Okay, now since different bacteria have different incubation period, so you can design the time intervals according to their incubation period. Okay, now in step 3, switch on the spectrophotometer at least 20 minutes before taking the absorbance. So it is highly recommended to switch on the spectrophotometer before 20 minutes. Now in step 4, withdraw 2 ml of the broth culture at every interval and measure the absorbance in the spectrophotometer at 610 nanometer wavelength. Okay, so at every interval what you do, you take 2 ml of the broth culture from your culture and you put it back in the incubator again, and you measure the optical density using blank. So what is blank? It is the uninoculated growth medium where the culture is not given. Simply it is a medium only. It's a media. So therefore it is taken as a blank. So using this optical density is taken against blank. Now here you will see that the 610 nanometer is written. Now different wavelengths are there for different bacteria. Okay, so different bacteria have different types of wavelength in which they give their absorbance. Now most of the bacteria, they are generally measured at a wavelength of 600 to 610 nanometer. You need to keep that in mind. Okay, so most of the bacteria are measured in 600 to 610 nanometer wavelength. However, there are some of the bacteria who have the different wavelengths at which their optical density need to be measured. Okay, so here's a note to set the absorbance or OD of the spectrophotometer to 0 using blank, as I said earlier also. So you need to set this optical density using the blank to 0, and after that only you measure your microbial culture against this blank. That way you will get the correct and precise optical density. Okay. And last, step 5, prepare growth curve by plotting the graph in terms of absorbance against time. Okay, so let us see how we will see our results. So this is the y and x axis as the time in hours and y axis as the absorbance at 610 nanometer OT. This is also referred to as OT, absorbance. Okay, now we will get somewhat this type of growth curve. This is for E. coli only. So since we have taken as an example of E. coli, so this is the growth curve of E. coli. Now, what type of growth curve is this? We have learned in our previous one. This is a sigmoidal growth curve, right? So this is the sigmoidal growth curve. Now here, if we try to find out which phase is what, so here this is the lag phase, this is the exponential phase, this is the stationary phase, and this is the death phase or decline phase. Okay, so this is how the growth curve is measured. You put every point, every absorbance at different graph at the different points of your graph. You can also do it in your Excel sheet, and you can also do it using your graph. Okay, so if you want a video on how to make a growth curve in an Excel sheet, put the comment down below. And here in the graph you can point the absorbance, the optical density which you have taken in differently, and then you can connect the dots which will give you somewhat this type of curve. Okay, so this is all for today's video. I hope you have understood this video. If you have any queries, don't forget to comment down below. If you want the full details of this video, don't forget to follow our LinkedIn page. The link is in the description. And if you have liked this video, give it a thumbs up. Do share and subscribe to our channel for more such related updates. And I'll see you in the next video.